students, this is Ankita. Welcome to Global Online Needs. So in today's session, we are going to discuss about the prokaryotic cell. Yes, so let's start with the session. What is prokaryotic cell, student? First of all, they have no membrane-bound nucleus and the organelles. Organelles, yes. Next, what they include prokaryotic cell? क्या include करता है students? They include bacteria, blue-green algae, mycoplasm, and PPLO. What they include? Bacteria, BGA, that is blue-green algae, mycoplasma, and PPLO, that is pleuropneumonia-like organisms. How are they? They vary in shape and size, and they are generally smaller and multiply more rapidly than the eukaryotic cells. This you can see in this diagram. This is bacteria. Again, this is bacteria. This is cyanobacteria, and this is mycoplasma or the PPLOs images. Yes. Next, it bacteria has four basic shapes. Yes. What the bacteria has? It has four basic shape. It is if it is rod-like, it is called as bacillus. Rod-like असे. असे जर बॅक्टेरिया असतील तर त्यांना काय म्हटलंय स्टुडंट्स? बॅसिलस. रॉड-like. Yes. Coccus, spherical, circular shape, spherical still bacteria, so it is called as coccus. Vibrio, that is comma shaped, comma सारखे bacteria still, so it is called as vibrio. And spiral, also called as spiralium. Yes, it is spiral in shape, like this. Yes, so it is called as spiralium. So the bacteria are of four shape: bacillus, coccus, vibrio, and spiralium. You can see in this diagram it is rod shape, it is called as bacillus. Spherical shape, it is called as coccus. This is comma shaped vibrio, and this is spiral like shape called as spiralium. Yes, now we are going to see about the envel prokaryotic cells and the organelles. Yes, it has cell envelope. Yes, prokaryotic cell को क्या है? Cell envelope. Then is the mesosome and chromatophores. Next is the nucleoid, flagella, pili and fimbriae, ribosomes and inclusion body. आता हे सगळे structures आपण one by one detail मध्ये बघणार आहोत. So please students draw along with me. Yes, we will draw the structure of a rough structure of the prokaryotic cell. Yes, so let's start. So draw along with me, students. A rough structure of the prokaryotic cell. Now this is the cell wall. Now we will use different ink for, drawing the plasma membrane. Students माझ्यासोबत draw करा तुम्ही पण. So that तुमच्या लक्षात राहील. Yes. Now we will draw the glycocalyx. Okay. हे आपल्या अंडरस्टँडिंग साठी ड्रॉ करतोय स्टुडंट्स आपण. येस, पिक्टोरियल डायग्राम आहे, बट फॉर आर बेटर अंडरस्टँडिंग वी आर ड्रॉइंग दिस. आणि ड्रॉ केल्याने आपले ते जास्त लक्षात राहतील, एकदम परफेक्ट. येस, फॉर दॅट पर्पज आपण ड्रॉ करतोय. आधी सगळे ड्रॉ करून घेऊ आपण आणि मग वन बाय वन वी विल लेबल इट. ओके, नाव वी विल ड्रॉ nucleoid, ribosomes, हे कशात आहे सगळं फ्लुइड? इट इज इन द साइटोप्लाझम. येस, this is the plasmid. Next, जास्तच इरेज झालं, एक मिनिट. ओके, again the same problem. स्टुडंट्स, मी आता असंच ड्रॉ करते, बट यू हॅव टू इरेज द टू पार्ट्स इन बिटवीन. ओके, आता फॉर अ इन्स्टंट आय विल ड्रॉ लाईक दिस, बट तुम्ही व्यवस्थित ड्रॉ करा. येस स्टुडंट्स, so here comes the body, that is mesosome, and here comes the chromatophore. Okay, then इथे छोटे छोटे granules present है, that is called the inclusion body. Then we will draw the pili, the fimbriae, the flagella. What else is remaining? Okay, so we will name it one by one. सगळ्यात पहिले, this is the rough structure of prokaryotic cell. Yes, it is the rough structure of prokaryotic cell. I hope you have drawn, students. Yes, आता naming करो. This red lining दिसतीये, it is nothing but the cell wall. Yes, this blue lining दिसतीये, it is plasma membrane. काय है? Plasma membrane. आणि ही ग्रीन कलरची डॉटेड लाईन दिसतीये, it is glycocalyx. Okay, what it is? It is nothing but the glycocalyx. Yes, together ह्याला काय फॉर्म करतात? This together forms a covering called as cell envelope. Yes students, next, this cell is floating in the cytoplasm. कशात float करतोय cell? In the cytoplasm. Yes, this is the pili. What it is? Pili. Next, these are the inclusion bodies. आता हे याचे सगळे डिटेल मध्ये आपण स्ट्रक्चर, फीचर, फंक्शन बघणार आहोत. ओके, This is a nucleoid. Nucleoid मध्ये काय असतं students? DNA. Yes. Next we will see this is the chromatophore. काय है? Chromatophore. Yes. याला काय म्हणतात? This hair-like structures called as bristle-like structures called as fimbriae. Yes. This is flagella. Right? Flagella कशाला use करतो? For locomotion. This body is mesosome. Okay? This is the plasmid. Is it clear, students? आता काय राहिलं? Yes, this is remaining. This and this. This is nothing but the ribosome. काय है? Ribosome. कुठलं रायबोसोम प्रेझेंट आहे प्रोकॅरियोटिक सेल मध्ये? 70S रायबोसोम. येस. इज इट ऑल क्लिअर? काय काय बघितलं आपण? फर्स्ट ऑफ ऑल द सेल एनव्हलप, ग्लायकोकॅलिक्स, प्लाझ्मा मेम्ब्रेन अँड सेल वॉल काय करतात? सेल एनव्हलप बनवतो. Then we saw the cell is floating in the cytoplasm. Then we saw the genetic material that is DNA, that is nucleoid. Then आपण रायबोसोम बघितला, क्रोमॅटोफोर बघितला, प्लाझमिड, फ्लॅजेला, फिम्ब्रे, inclusion body, mesosome. Yes. So आता one by one आपण ह्याचे सगळे characteristics, features and functions बघणार आहोत. Okay. Now let's start with the cell envelope. Cell envelope काय student? It is a chemically complex protective covering. कसं आहे हे? Protective covering आहे. Cell envelope ला काय students? It has a protecting covering. It is made up of three tightly bound layers: glycocalyx, cell wall and plasma membrane. बरोबर आहे? Glycocalyx सगळ्यात outer आहे. The middle is the cell wall and the innermost is the plasma membrane. Yes, students. So let's see what is glycocalyx. So glycocalyx is the outermost layer. काय glycocalyx? Outermost layer. It is made up of कशाने made आहे? Made up of glycocalyx is made up of mucopolysaccharide. Mucopolysaccharide. Next is काय feature आहे glycocalyx चा? Sir, it is composed. It has different composition and the thickness varies. बरोबर आहे? Glycocalyx differs in the composition. Suppose जर thick असेल, yes, if the covering is rigid, that means if it is thick, it makes capsule, making capsule, okay, called as capsule. Whereas जर त्याची loose sheet असेल, तर it is called as slimy layer. Is it clear, students? Glycocalyx is the outermost layer of the prokaryotic cell. It is made up of mucopolysaccharide. Its composition differs and thickness also. If it is rigid and thick, it is making capsule that is thick and tough. If it is making loose sheet, it is called as slimy layer. Yes. Now, what is the function of glycocalyx? क्या function है glycocalyx का? Helps in adhesion. Yes. It helps in adhesion. Next, prevents loss of water. Prevents loss of water. And next, अजून काय करतं glycocalyx? Gives protection. Yes, फ्रॉम द होस्ट इम्यून सिस्टम. म्हणजे त्याचं काय काम आहे? 3 काम आहेत. 3 फंक्शन आहे स्टुडंट. रिमेंबर, it helps in adhesion, it prevents loss of water, and it gives protection. So this was all about the glycocalyx. Now let's see the cell wall. Cell wall, the middle layer. बरोबर आहे? आता cell wall कशाने made आहे? It is made up of peptidoglycan. Yes? Cell wall, the inner middle layer. First of all, काय cell wall? It is middle layer. It is made up of peptidoglycan. Peptidoglycan. Yes. Next काय? Cell wall चं काय काम आहे student? It determines the shape of the cell. Yes. Determines the shape. अजून काय काम आहे स्टुडंट्स सेल वॉलचं? इट गिव्स स्ट्रक्चर. बरोबर आहे? स्ट्रक्चर प्रोव्हाइड करते. गिव्स स्ट्रक्चरल फ्रेमवर्क म्हणजे स्ट्रक्चर प्रोव्हाइड करते. अँड इट ऑल्सो प्रिव्हेंट्स द सेल फ्रॉम बर्स्टिंग. येस. It prevents the cell from bursting. Prevents it prevents the cell from bursting. बरोबर आहे? Now, is the cell wall seen in all cells? No, it is seen in all prokaryotes. Yes, it is a middle layer seen in all prokaryotes. It is made up of peptidoglycan. It determines the shape of the cell. It gives structure to the cell, and it also prevents the cell from bursting. Yes, this was all about the cell wall. Next is the plasma membrane. It is the innermost layer of the cell. बरोबर है? Innermost layer of the cell. How is this cell, students? It is selectively permeable. Selective membrane का? Because it allows everyone to let in? No. काही specific substances ला ती आत येऊ देते. Inside the cell. So that's why it is called as selective membrane. It interacts with the outside the cell. Interacts with outside the cell. Yes, students. Next guy. What it is next? It is same as the eukaryote. Clear? What is the plasma membrane? It is the innermost layer of the cell. It is selectively permeable membrane or semi-permeable in nature because it allows a specific substances to let in. It interacts with the outside of the cell, and it is similar in structure to the eukaryotes. So here the cell envelope is complete. Cell envelope has 3 layers, that is glycocalyx, cell wall, and the plasma membrane. Glycocalyx is called as the outermost layer. Cell wall is the middle layer. Plasma membrane is the innermost layer. So here we saw about the cell envelope and their functions. Now we will see the gram positive and gram negative stain. So Christian Gram, he developed the staining method. Who developed? It will come as a MCQ question, student. Christian Gram developed the staining method. On the type of the cell envelopes and response to the gram staining, bacteria are of 2 types. कौन से bacteria? 2 type के bacteria, gram positive and gram negative. जर त्या bacteria take up and retain the gram stain, it is called as gram positive bacteria. जर त्या बॅक्टेरियाने स्टेन रिटेन नाही केला तर इट इज द ग्राम नेगेटिव्ह बॅक्टेरिया. इज इट क्लिअर? Now we will see about the mesosome and chromatophore, that is the membranous structures. आता हे mesosome and chromatophore कुठल्या membrane ला आहे student? Is it to cell wall, glycocalyx or plasma membrane? It is to the plasma membrane. So this is the modification of, we will see the modification of plasma membrane. Yes, प्लाझ्मा मेम्ब्रेन. सी स्टुडंट्स, हे आपण आपल्या डायग्राम मध्ये बघितलं होतं. येस, a rough diagram which we draw. Yes, we have completed the cell envelope. Now we are seeing the mesosome. Yes, the inward, काय ह्याला inward structure आहे. Plasma membrane is making the inward structure called as mesosome and chromatophore. So in this diagram, this is the mesosome. Mesosome also called as lamellosome. Yes, it is also called as lamellosome. Now, lamellosome includes, see, it is formed by the infolding of plasma membrane. Infolding also called as the inwarding of the plasma membrane. Yes, students. Next is, it is showing vesicles, tubules and lamella. Mesosome काय दाखवतं? Mesosome shows, or mesosome includes vesicles, the next is the tubules or tubules and the lamellae. Yes. Now this mesosome has certain function. Yes, काय function है mesosome चं? First of all, it helps in the cell wall formation. Cell wall form करण्यासाठी कोण help करतं student? Mesosome. It helps in respiration. कशासाठी help करतं? Respiration. And next is the DNA replication. DNA replication same as to the binary fission. म्हणून इथे दिलंय for distribution of chromosomes to the daughter cells. So what are the functions of mesosome? It helps in cell wall formation, it helps in respiration, it helps in DNA replication. बरोबर आहे? And also to increase the surface area of the plasma membrane and the enzymatic content. Yes student, आता हे mesosome जर आपण कंपेअर केलं युकॅरियोटिक सेल बरोबर तर कुणासारखं हे ऍक्ट करतं? It acts as a mitochondria. बरोबर आहे? Because mitochondria मध्ये काय होतं? Respiration होतं students. Yes. So it acts as a mitochondria of the eukaryotic cell. It acts as a mitochondria of eukaryotic cell. Yes. Mesosome acts as a mitochondria of the eukaryotic cell. Clear mesosome? Now we will see the chromatophore. Chromatophore पण काय? की it is the infolding किंवा inward काही plasma membrane मध्ये change झाला त्याच्यामुळे chromatophore form झालं. It is not present in all cells. Yes, this chromatophore is not present in all cells. Is it present? No. Next, they are the pigment containing membranous enfolding in some prokaryote. म्हणून सगळ्या cells मध्ये आहेत का ते? नाही. Example is cyanobacteria. Yes. Now what is the function of this chromatophores? काय करतात chromatophores? It performs photosynthesis. काय करतात हे chromatophore? Performs photosynthesis. Example काय आहे याचं photosynthesis चं? Example is photosynthetic bacteria. Example is photosynthetic bacteria. Yes. आता हे क्रोमॅटोफोर जर आपण कंपेअर केले स्टुडंट्स युकॅरियोटिक सेल बरोबर, तर कुणासारखे हे ऍक्ट करतात? येस, इट ऍक्ट ॲज अ क्लोरोप्लास्ट. कुणासारखे ऍक्ट करतात? क्लोरोप्लास्ट ऑफ द eukaryotic cell. Yes, is it clear? Chromatophores clear झाले? Yes, now we will see the nucleoid. Nucleoid काय student? It is the genetic material of the cell. Yes, nucleoid कसा आहे? It is naked. What is the characteristic of the nucleoid? It is naked. No histone proteins is present. No histone protein present. How it is nucleoid कसा form मध्ये? It is circular. Circular genomic DNA, single chromosome or a genetic material and protein. बरोबर है? It has polyamines that helps in DNA packaging. First of all, it is form of non-membranous. It is naked. It is circular genomic DNA. No histone protein is present. It is circular in nature, and many bacteria have small circular DNA plasmid outside the genomic DNA. It gives some unique phenotype characters, that is resistance to antibiotics to bacteria. म्हणजे काय, हे जेनेटिक मटेरियल सर्क्युलर डीएनएच्या व्यतिरिक्त काही असे बॅक्टेरिया आहेत ज्यांना स्मॉल सर्क्युलर डीएनए प्रेझेंट आहे आउटसाइड द जेनोमिक डीएनए, त्याला आपण काय म्हणतो स्टुडंट? दॅट इज कॉल्ड ॲज प्लाझमिड. व्हॉट इज कॉल्ड? That is called as plasmid. Yes, so it is all about nucleoid. Next comes the flagella. Yes, आपण बघितलं flagella काय? It is a thin thread like structure, right? These are the thin filamentous extensions of the cell. काय help करते flagella? For motility. It helps in motility. Yes. Next काय? कशाने बनली आहे flagella? It is made up of flagellin protein. Yes. No 9+2 arrangement is seen in this. No 9+2 arrangement seen in this. Yes, it has 3 parts. Bacterial flagellum ला काय आहे? 3 parts आहेत. Which are they? Let's draw. Yes, this is the basal body. This is the hook, and this is the filament, which is the largest part. Yes, filament that is long and large. Okay, so the filament is the longest portion and extends from the cell surface to the outside. Is flagella clear, students? Kai flagella? It is the longest thin filamentous extension from the cell wall. It helps in the motility of the cell. No 9+2 arrangement is seen in this. It is made up of flagellin protein. Bacterium flagellum has 3 parts, that is filament, hook and the basal body. The filament is the longest portion and that extends from the cell structure to the outside. Next comes the pili and fimbriae. Right? Pili कसा आहे? It does not help in motility. No role in motility. Okay? No role in, no role in motility. Yes. How it is? It is long tubular. कसा है? Long and tubular structure, and it is made up of special protein that is pylene. Pylene protein से made है. It helps in conjugation. Helps in conjugation. Yes, students. So, pili are the long tubular structures. They have no role in motility. They are made up of special protein called as pylene, and it is helping the pili helps in conjugation. Yes. Now, fimbriae are the small bristle-like fibers. You can see here. आपल्या diagram मध्ये आपण असे असे draw केलेत, right? These are the small bristle-like fibers. It helps in the attachment of the bacteria. So what is the function? Helps in the attachment. एक तर substratum. Yes, एक तर substratum attachment ला help करता. Otherwise to the host. Yes, help to attach the bacteria to the rocks and streams and the host tissues. So these was all about the pili and the fimbriae. Yes, next comes the ribosomes. Ribosomes ला काय म्हटलंय students? It is called as a protein factory. Ribosome is called as a protein factory. Yes, it helps in the protein synthesis. काय काम है ribosomes चं? It helps in protein synthesis. कुठे असतात रायबोसोम? They are associated with the plasma membrane of the prokaryotes. What is the size? Size is 15 to 20 nanometer. Now these ribosomes are made up of two subunits. Two subunits. Yes. काय दोन subunits? First of all, 70S type of ribosome is seen in the prokaryotic cell. They have two subunits. It is called as 30S and this is called as 50S. Right? 30S is the smaller subunit and 50S is the larger subunit. S is the sedimentation coefficient, a measure of density and size. Yes? Now these ribosomes have the special functions. Ribosomes are the site of translation, that is protein synthesis. म्हणजे first function काय झाला? Protein synthesis. Yes. Now this is a PYQ question, students, that several ribosomes may attach to a single RNA. काय होतं when many of the ribosomes are attaching to the mRNA? This it is called as polyribosome. You can see in this diagram. See, हा काय आहे? mRNA. याला किती most of the ribosomes are attached. So this is called as polyribosome. Polyribosomes or polysome. Yes, these ribosomes of a polysome translate the mRNA into the proteins. What does they do? They translate the mRNA into proteins. Next comes the inclusion body. आता inclusion body, how it is? It is non-membranous, right? Granules आपल्या diagram मध्ये आपण असे काढलेत. It is the granule-like structure. You can see in this diagram, granules-like structures आहेत. काय करतात? What is the role of this inclusion body? They reserve or store food. काय करतात inclusion body? They store food in the prokaryotes. They are the membraneless structures. म्हणून यांना काय म्हटलंय? Membraneless structures, so called as non-membranous. Yes. Example काय आहेत याचे? Phosphate granules, synophasian granules, glycogen granules and the gas vacuoles. Yes, gas vacuoles are found in the blue, green and purple and green photosynthetic bacteria. कुठे भेटल्यात gas vacuoles? Gas vacuoles are found in the blue, green, and purple and green photosynthetic bacteria. So here we complete with the prokaryotic cell. So let I give you quick recap of it. Coming back to our diagram, see, we have completed the cell envelope, we have completed the mesosome, then we saw about the chromatophore, we saw about the nucleoid, we saw about the flagella, we saw about the fimbriae, we saw about the pili, we saw about the ribosome. Yes, inclusion body. Now, what is plasmid? Yes, what is plasmid? Is it present in all bacteria? Let's see. No. Now we will see the plasmid. Yes, plasmid. It is not present in all bacteria. Yes, it is an extra chromosomal DNA. Extra chromosomal DNA. Right? आपण वाचलं तिथे की it is different from the genetic material. It gives the certain phenotype characters. काय घेतं ते? Phenotype characters देतो. Phenotypic character. Next, it is antibiotic to resistance. Antibiotic resistance. Yes, and it is used for the bacterial transformation. Bacterial transformation. Yes, students. Is it clear? So let's revise once what we saw. Prokaryotic cells are the doesn't have membrane-bound nucleus and organelles. They include bacteria, blue-green algae, mycoplasm, and PPLO. They vary in size and shape. The bacteria has four basic shapes. Yes, bacteria have four basic shapes. Bacillus rod-like, coccus spherical, comma shaped is called as vibrio, and spiral is spiralium. You can see in this diagram, Bacillus spherical, vibrio, and spiralium. Next, we saw about the prokaryotic cell. And prokaryotic cells have cell envelope, mesosome, chromatophores, nucleoid, flagella, pili and fimbriae, ribosome, inclusion body. We saw each of its characteristics and functions. Yes, then before going to that, we have drawn a rough diagram of the prokaryotic cell. Yes, what is an envelope? It is a chemically complex protective covering. It has three tightly bound layers, that is glycocalyx, cell wall, and plasma membrane. Glycocalyx outermost layer. Yes, it varies in thickness and composition. If it is rigid, it is called as capsule. If it is loose sheet, it is called as slimy layer. It is made up of mucopolysaccharide. The functions of glycocalyx is hydration, prevents loss of water and protection. Next comes the cell wall. It is the middle layer of the cell. It is made up of peptidoglycan. What the cell wall does? It determines the shape and the structure of the cell, and it also prevents the cell from bursting. Right? Next comes the plasma membrane. It is the innermost layer of the cell. It is the selectively permeable membrane, or also called as semi-permeable membrane, as it allows specific substances to let in. It also interacts with the outside the cell. Then we saw about the gram-staining bacteria. Depending on the two types, gram-positive, if they take up and retain the gram stain, they are called as gram-positive. If they do not retain the stain, they are called as gram-negative bacteria. Yes, then we saw the mesosome and chromatophores. These mesosome and chromatophores are the modification of the plasma membrane because they are formed by the inwarding or the infolding of the plasma membrane. Yes, mesosome also called as lamellosome. It includes vesicles, tubules, and lamellae. The function of mesosome is the formation of cell wall, respiration, DNA replication, and to increase the surface area of plasma membrane. If we compare the mesosome to the eukaryotic cell, it acts as a mitochondria of the eukaryotic cell. because respiration is seen in the mitochondria. Yes, the next enfolding is of the chromatophore. These are the pigment containing membranous enfolding in some of the prokaryotes, so they are not present in all the cells. It performs photosynthesis. Example is photosynthetic bacteria. If we compare this chromatophore with the eukaryotic cell, it acts as a chloroplast of the eukaryotic cell. Yes, because photosynthesis is taking place. Next is the nucleoid. Yes, it is present in a circular form. It is naked. It is non-membranous. Yes, no histone protein is present in it. And as we saw that plasmid, so many of the bacteria have circular DNA, that is plasmid, that is present outside the genomic DNA. Yes, what is this plasmid? It is for unique phenotypic character and the bacterial transformation and also the antibiotic resistance. So here that was about nucleoid. Next we saw the flagella. It is a long, thin, like thread-like structure. It is made up of flagellin protein. No 9+2 arrangement is seen in this. What is the role of this flagellum or the flagella? It helps in locomotion, that is motility of the cell to move from one point to another. This flagella is divided, has three parts, that is basal body, hook and the filament. This filament is the longest portion and extends from the cell surface to the outside. Yes, next we saw the Pili and Fimbriae. What is pili? They have no role in motility. How is the structure? It is long and tubular and it is made up of a special protein called as pili, which helps in conjugation. Now this fimbriae are the small bristle-like fibers. Yes, what is the role? They attach the bacteria to rocks in streams and to the host tissue, substratum or the host tissue. Next comes the ribosome. Ribosomes are called as the protein factory as it is helping protein synthesis, right? They are associated with the plasma membrane of the prokaryotes. In prokaryotes, 70S type of ribosomes are seen. Yes, what is the size of the ribosome? 15 to 20 nanometer. They are made up of two subunits, 50S and 30S, right? 30S is called as the smaller subunit and 50S is called as the larger subunit. What is the function? Protein synthesis, and it is a PYQ question that when a single mRNA core several ribosomes are attached, it is called as polyribosome or polysome. Yes. Next we saw about the inclusion body. What is the role of the inclusion body? They are present as the small granules, non-membranous. Yes, inclusion body store the food of the prokaryotic cell. Yes, and they are freely in the cytoplasm of the prokaryotic cell. There is no fixed location. They are floating in the cytoplasm and they store food. Example are phosphate granules, synovial granules, glycogen granules, and gas vacuoles. These gas vacuoles are found in the blue, green, and purple and green photosynthetic bacteria. So, students, here we complete with the prokaryotic cells. Yes, thank you.